The definition is identifying bacteria species based on the physical characteristic of both their colony and their individual cells. There are few differences between colony and cell morphology, such as definition, shape, observation, and usage. The definition of colony is visible cluster of microorganisms, typically bacteria or fungi that grow on solid surface, while the cell morphology is study of the shape, size, structure, and arrangement of individual cells. The shape of colony is round, irregular, and filamentous, and the cell morphology is cocci, bacilli, and spirilla. The observation of colony is with naked eye on an agar plate, and the cell morphology is under a microscope. The usage of colony is often used as the first step in identifying bacteria, indicating possible species, and the cell morphology is used for more detailed identification, especially for gram staining and cell structure. The principles are based on the observation and analysis of the physical characteristic of bacteria, colonies, and individual bacteria cells. The colony morphology gives initial clues about the bacteria species based on visible growth pattern, while the cell morphology provide more detailed definitive identification by examining the bacteria cell structure under the microscope.
Now, let's move on to the purpose of identification of bacteria using colony and cell morphology. There are several purposes of doing this experiment. The first one is classification and taxonomy. This is because it assists in organizing bacteria into broader categories for identification. Secondly, differentiation of species. It's in distinguishing between different bacterial species based on colony appearance and cell shape. Lastly, assessing growth condition. This is because it provides clues about the bacteria's preferred growth environment, such as aerobic and anaerobic condition.
Now we take a look at colony morphology steps. The first one is we need to prepare the culture. Take a sample and streak it on an agar plate using aseptic techniques. The goal of streaking is to isolate single bacterial colonies. Each colony typically arises from one bacterial cell. The second one is we need to incubate. Place the plate in an incubator set to the optimal growth temperature for the bacteria. Incubate for 24 to 48 hours or longer, depending on the bacterial species. The third one is we need to observe colony morphology. After incubation, examine the plate to identify distinct bacterial colonies. Look at the differences in size, shape, color, texture, margin, and elevation. Now, I am going to talk about the cell morphology steps. First one, we need to prepare a smear. Pick a small sample from a bacterial colony using a sterilized inoculating loop. Place it on a clean glass slide. Mix it with a drop of water if needed and spread evenly. Air dry the smear completely, then heat freeze it by passing the slide quickly over a flame. Heat freezing kills bacteria and adheres them to the slide. Next, we need to stain the cell. First one, we need to apply a staining reagent. There are three types of stain. The first one is gram stain. It used to differentiate bacteria into gram positive, which is purple, and gram negative, which is pink. Next is simple stain. It highlights bacteria size, shape, and arrangement without differentiation. The third one is special stains, which highlights features like capsule, spores, or flagella. For the third step, we need to do a microscopic examination. We need to observe the stain slide under a microscope, starting with low magnification and moving to high magnification, which is oil immersion at 100x if often required. Note the bacterial shapes such as spherical, rod shape, or spiral, and arrangement such as chains, clusters, or pairs.
Now, I want to tell you the benefits of doing this method. Firstly, it quick and simple process. The technique are relatively fast, providing preliminary identification without requiring advanced equipment. Secondly, cost effective. These methods are inexpensive compared to molecular techniques, making them accessible for many laboratories. Thirdly, facilities large-scale screening. Morphological identification can be used for screening large numbers of samples in public health or research setting, helping track bacterial strains. Lastly, provides immediate clues. Colony and cell morphology can quickly suggest potential bacterial species, guiding further diagnostic tests and treatment.